Hi everyone, thanks for joining us today. My name's Lucy. I work on our emerging technologies portfolio here in the UK, helping to keep you up to date with the latest exciting new tools and technologies from Promega R&D. In this presentation, I'm going to take you on a whistle-stop tour of our Lumit platform, how it came about, how to build your own assay, and an overview of the pre-built assays we have available for you to use right now. This is a very brief overview. We do have plenty more data we can share, so if you find any particular area interesting, please do get in touch. By the end of this presentation, I hope you will have an idea of how the technology works, as well as some ideas on how to use it to progress your own research. Lumit Technology is based on our NanoLook luciferase technology, which some of you may already be aware of. NanoLook is a very small, 19 kilodalton, molecularly engineered luciferase molecule that is extremely bright. In fact, it's 100-fold brighter than your standard firefly luciferase. Promega R&D adapted this enzyme to create a split luciferase complementation system called NanoBit. The enzyme is split into two parts. Small bit, which is just 11 amino acids, and large bit, which is 17.6 kilodaltons. These two component parts have been evolved to have very low affinity to one another, so on their own you have no signal. However, when the two bits are fused to two proteins that interact with one another, the proteins interacting brings the small bit close to the large bit and allows the reconstitution of nanobit luciferase, generating a light signal in the presence of the substrate that is proportional to the number of proteins interacting. Lumit Technology uses the same split luciferase technology, but in this case, instead of tagging proteins of interest with large bit and small bit, antibodies are tagged. If two different antibodies that bind to the same analyte are tagged, then in the presence of both analyte and substrate, light is produced that is directly proportional to the amount of analyte in a sample. Lumit immunoassays are designed to be easy to use and can be performed directly on cells and medium with no need for transfer or wash steps. They're very fast, with 30 to 120 minute assay times. They're very sensitive, but also work over a broad dynamic range, minimising the need to dilute high concentration samples to bring them into the assay window. They use a standard luminometer, so there's no need for expensive specialist equipment, and they're amenable to high throughput applications. I'm now going to walk you through the three different assay options we have currently available. Firstly, in the direct configuration, primary antibodies themselves are labelled with the bits. This allows you to test multiple different antibody pairs and minimises the number of components required. In the indirect assay format, we use labelled secondaries. This allows you to use as many off-the-shelf unlabelled antibodies as you like, so long as they've been raised in different species, for example, mouse and rabbit or rabbit and goat. I will describe the use of this assay system to interrogate intracellular signalling nodes. Finally, we have the competitive binding scenario, in which a tracer that binds to the antibody is labelled rather than two antibodies. This allows you to add unlabelled analyte that competes for binding with the tracer. There isn't time to go into detail in this talk, but we have developed an FcRn binding assay using this approach. R&D have developed a number of direct assays to various cytokines and some key metabolites. In this section, I'll show you what's available at the moment and use interferon gamma as an example of how to build a direct immunoassay. As an introduction, cytokines are numerous small proteins that are released from immune and non-immune cells. They signal through cell surface receptors in order to exert their biological effects. They elicit critical inflammatory and immunomodulatory roles within the body. They are, of course, targets for cancer and inflammatory disease and are frequently measured in cell culture models and other biological samples. Insulin and glucagon are key target analytes measured in metabolism research. Secreted from beta and alpha cells of pancreatic islets respectively, the two hormones are vital for regulating glucose levels in the body. R&D have already developed a number of off-the-shelf direct immunoassays as listed here. We can also build you an assay as part of our custom assay services. Please reach out to discuss further. If you want to build your own assay, we have the tools to help you do that too, and I'll show you how using interferon gamma as an example in the next few slides. In order to build a Lumit assay, the first thing you need to do is label your target antibodies. Here you can see a gel where we have interferon gamma antibodies labelled with the Lumit tags. Here you can see 7 different antibodies labelled with small bit and with large bit, and you can see the ladder effect compared with the unlabelled antibody here on the left. We're typically looking for 3 to 5 labels per antibody, but there's some tolerance in the system, so it doesn't have to be exact. This labelling and gel analysis stage typically takes just 2 days. Now that you have your labelled antibodies, you want to test them to see which pairs perform the best. To do this, you set up an array like the one shown here, with small bit along the columns and large bit along the rows, using a fixed concentration of analyte and 2 plates with no analyte to give you the background. From this, you can calculate the signal's background and generate a heat map to identify the best antibody pairs. From this, you can quickly see a number of hotspots identifying good antibody pair combinations. This rapid screening can be completed in just one day. Then, on day 2, you can go on to test the best antibody pairs against standard curves or against cells to detect native proteins released from the cells in order to optimise the concentrations of the labelled antibodies selected. On the left, we have the standard curve of human interferon gamma. You can see from this graph it's extremely bright. We can detect several million RLUs at high concentrations, but the assay is also very linear. Down at 6 picograms per mole, we still have a signal to noise of 10. This is a big advantage of this system. It gives a very broad dynamic range, which enables you to analyse a wide range of analyte concentrations without the need to dilute. On the right here, you can also see the excellent sensitivity and linear range for several human cytokines, indicating that this assay is suitable as a universal tool to detect a wide range of analytes. This is what the standard workflow looks like. It's very straightforward. You plate your cells and a standard curve in a standard 96-well plate, treat them as desired, and then add your labelled antibodies. Wait 60 minutes for the complex formation to complete, and then add the detection reagent and read. There are no washes, no transfers, and the whole assay can be completed in around 70 minutes. You can also use the direct assays to detect insulin secretion. In this graph, the INS1 cells were treated with increasing concentrations of glucose as plotted on the x-axis. After 60 minutes, 10 microliters of supernatant was removed and assayed with the Lumit insulin immunoassay in 384-well plates. You can see here increasing release of insulin with increasing concentration of glucose as expected. You might be wondering how exactly you go about labelling your primary antibodies. Well, there are 2 simple ways of doing this. Either you can purchase a DIY kit yourself to label your own antibodies, or you can engage us as a service provider to label your antibody for you. In either case, first the primary antibody of interest are labelled with a chloralkane linker, which binds to the free amines and lysines on the protein. Then the halotag fusion proteins are added, and the halotag domain binds covalently to the linker to conjugate halotag protein to the antibody, which you can then analyse on a gel. In summary, the Lumit cytokine and metabolism assays utilise the direct label approach to quantify the amount of cytokine or metabolite present in the sample. They are simple, homogeneous assays that can be performed directly on treated cells with no wash or transfer steps required and a rapid protocol that doesn't require any specialist equipment. I've shown you that the Lumit assay has excellent sensitivity and a broad linear range. In addition, we've done experiments in 384-well format to demonstrate that these assays are miniaturizable for high throughput applications. We have several assays available or ready to purchase and more are in development. We can also support you in building your own assays by labelling your own antibodies. Next, I want to talk to you about the indirect Lumit method, which uses labelled secondary antibodies purchased from Promega and your own unlabelled primary antibodies. This assay can work with any commercial source of antibody pairs, provided the 2 antibodies are from different species, for example, mouse and rabbit or mouse and goat. This assay system we have called our Lumit Immunoassay Cellular system, and we've used it to quantify phosphorylation and degradation events in order to probe intracellular signalling events in unmodified cells. In this example, we're interested in measuring both phosphorylated and total I-kappa-B alpha levels. This signalling pathway is particularly interesting because upon treatment with TNF-alpha, I-kappa-B alpha is phosphorylated, which in turn leads to its degradation. So first we need to find a pair of antibodies that will bind to phosphorylated I-kappa-B alpha. To do this, we screened a number of commercially available antibodies and plotted the fold response of treated versus untreated cells in a heat map format. Here you see the results from one pair, here from a second pair, and here a third. From this, you can see that the particular antibody pair combination and concentration that give the best fold response are these ones here. So these are the ones that we chose to work with. Here's the workflow of a Lumit assay using the antibodies chosen at the last stage. As you can see, this is a simple and rapid workflow requiring no medium removal, washes, or transfers. The green line here demonstrates that in non-treated cells, the total amount of I-kappa-B increases with cell number as you would expect. In contrast, in cells treated with TNF-alpha, the amount of protein is much lower, as you would expect from the biology. The second graph shows that if you block protein degradation using the proteasome inhibitor MG132, the amount of phosphorylated I-kappa-B alpha protein increases in TNF-alpha treated cells, shown in red, whereas in untreated cells it remains low, as you would expect. Promega R&D have now screened antibody pairs for more than 20 phospho and total proteins using 8 different cell types and continue to develop more protocols. We're making these application notes available on the Lumit homepage so that you don't need to work up these methods for yourself. The protocols will tell you where to buy the antibodies and what working concentrations we found best, as well as the correct Promega detection system to use and detailed protocol guidance. You can also develop assays for your own targets using the methods described, provided you have a pair of good antibodies raised in either mouse, rabbit or goat. In summary, the Lumit Immunoassay platform offers you rapid, no wash, no transfer assay formats with a large dynamic range, which can be performed directly on cells. It requires only a standard plate reading luminometer with no expensive equipment needed. It's scalable for high throughput use, and our platform includes both ready-to-use kits as well as reagents to create novel detection assays for yourself. Here you can see a summary of the off-the-shelf kits we have available right now. And here you will find the tools you need to build your own custom Lumit assays. Thanks for listening. If you have any questions, please do reach out either to myself or to your local Promega contact, and we'd be glad to help you.